Welcome to this live Q&A event hosted by Tinnitus Quest. Today is actually our first live event where we're together in the same room with our guests, live from California, where I have the pleasure of speaking with Dr. Susan Shore and Jon Pearson from Auricle, the company that is commercializing Dr. Susan Shore's tinnitus treatment invention. Now, this event has been highly anticipated as Auricle has been very sparse over the past few years with communicating anything publicly. But today they're ready to share with you everything related to the development of the device, the steps taken so far, and the road that's ahead to get this device to market for patients to actually use. But before we dive into that, let me tell you a little bit about Tinnitus Quest and why we organize events like this. First of all, we're not commercially affiliated or otherwise affiliated with Auricle. We're not being paid to host this event. In fact, we're covering all the costs ourselves, nor are we seeking to promote any particular company or treatment. Rather, we are seeking to bring news, information, and education to the tinnitus patient community. And we've already organized a series of live Q&As with leading researchers from around the world that you can find on our website. So really what we want to do is show you that progress is being made. There is hope. There are people, dedicated people, scientists, innovators, working on potential solutions for tinnitus. And that is not all that Tinnitus Quest does. We also fund research directly through our grants program. But rather than telling you about everything we do, let us show you in a short video. Tinnitus Quest was founded just over a year ago by a group of patients who were frustrated with the lack of progress in finding real solutions that could silence tinnitus. So we decided to take matters into our own hands. We now have some of the top scientists on our board, and we've raised enough money to be able to distribute $400,000 in research grants. We received 29 applications to our grants program. Each application represents a fresh new idea for how tinnitus could be silenced. We're in the process of reviewing these applications and we'll be ready to announce the winners by mid-December. But enough about Tinnitus Quest. Let's talk about the structure of this event. You will first hear Susan present all the science related to her device and the studies that were done so far. That will take about 30 minutes. Then Jon will talk about the commercialization of the device and the route to market, which will take about 20 minutes. After that, we'll move to the Q&A portion. We'll start with some pre-submitted questions. Over the past few weeks, we've been reaching out to a number of online and offline patient communities, and over 200 people have already submitted their questions. Some people have even submitted 2 or 3 or 4 questions, so we literally had many hundreds of questions to go through, review, categorize, and from that we came up with about 7 or 8 key questions that represent what the community most wants to know. After that, we'll move to the live submitted questions. So if you're tuning in live today, you will see on your screen a Q&A box where you can submit your question. You can already start doing that now, and you can also upvote other people's questions. So if you see a question that piques your interest, vote on it, and the questions that are most upvoted are more likely to be asked. Unfortunately, we probably won't have time to cover everyone's questions today, and I do apologize for that, but please do submit them. We will review all of them afterwards and follow up if we can. Now, let me introduce our speakers for today. Dr. Susan Shore is a professor emerita of otolaryngology, molecular and integrative physiology, and biomedical engineering at the University of Michigan's Kresge Research Hearing Institute. She's also fellow of the American Association for the Advancement of Science. She's known for uncovering how touch and sound pathways interact to produce tinnitus. And she developed a non-invasive bimodal stimulation approach to reverse those neural changes. She has authored over 70 scientific papers, mentored many scientists, and co-founded Auricle Inc. Jon Pearson is the co-founder and CEO of Auricle Inc. He joined Auricle in 2019 after a long career leading consumer health and medtech ventures, where he worked on product development, market entry, and global sales. He brings expertise in scaling early-stage medical technology companies, and under his leadership, Auricle secured licensing from the University of Michigan and advanced towards regulatory approval for the device. Now, let's move on to the presentations. As mentioned, you'll first hear from Dr. Susan Shore, followed by Jon Pearson. After both talks, we'll open up the floor for the Q&A session. All right, over to you, Susan.
Thank you, Hazel, for this wonderful invitation and the opportunity for Jon and I to address this wonderful audience. So I'm going to get started right away, and I'm going to tell you about from scientific discovery to Auricle. First, I have to tell you about my disclosures, funding that I've had over the many years that I was doing research in the lab, and also U.S. patent, and that I'm a co-founder and CSO of Auricle. So what is tinnitus? Tinnitus is the perception of sound in the absence of an auditory stimulus, as you all know. So because of that, if we want to investigate tinnitus in the brain, we have to measure spontaneous activity in a neural structure. And I'm going to get to that in a minute and explain what that is to you. And we need to look first at what the normal system does. And then we have to look and see what the system does after noise exposure and after tinnitus induction. So what is spontaneous activity? Here we have a schematic of spontaneous spikes over here. This is spontaneous activity in this neuron. And you can see that there are very few spikes. Most neurons in the central nervous system have spontaneous activity. And this is what you see in the auditory system in silence. So both of these neurons now are showing low spontaneous firing. But when you add a sound stimulation, for example, you're now looking at sound-driven spikes, and you can see that there are many more spikes. And this is typical of how a neuron in the auditory system will respond to sound. These are sound-driven spikes. Now, if an animal or a human has tinnitus, their spontaneous activity is going to look more like this. And so as scientists, we have to look at this activity and measure it and study it. And so that's partly what I'm going to be telling you about today. I'm going to be telling you about our preclinical studies, these are animal studies, and how these evolved to perform human trials. I'm going to tell you that fusiform cells of the dorsal cochlear nucleus encode tinnitus in their spontaneous firing rates and spontaneous synchrony. Fusiform cell synapses that receive somatosensory inputs are plastic, and I'm going to explain to you what this is. Fusiform cell plasticity is altered in tinnitus, and reversing the altered plasticity can treat tinnitus. And then I'm going to show you what we did in our clinical trials and what the human tinnitus sufferers say. So this is a schematic of the brain, and the position of the cochlear nucleus sits here. It's in the brainstem. It's what's called a cerebellopontine angle. And you can see the schematic here of the cochlea shows the auditory nerves coming in to connect with the cochlear nucleus. The cochlear nucleus is divided into a dorsal division, the dorsal cochlear nucleus, or DCN, and the ventral cochlear nucleus. Today we're going to talk only about the dorsal cochlear nucleus. So now we look at a simplified circuit of the dorsal cochlear nucleus. And we're going to focus in this talk on these cells called fusiform cells, abbreviated here FC. You can see in the simplified circuit that the fusiform cells receive auditory nerve input from the cochlea on these dendrites at the bottom of their cells. And they receive somatosensory input in these dendrites at the top of the cell. And this is important to remember for the rest of the talk because I'm going to keep talking about the somatosensory inputs and the auditory inputs to the fusiform cell. Another thing that's important to remember is that these fusiform cells send their axons to the central nervous system to more central stations, for example, the inferior colliculus and the medial geniculate body of the thalamus. So everything that's coded here in the cochlear nucleus is sent up to the rest of the brain so that the rest of the brain gets the signal from the fusiform cells. Now I'm going to show you what happens when we record spontaneous activity from these fusiform cells. And what this schematic here is showing is the dorsal cochlear nucleus and how the cells are aligned from low frequencies to high frequencies. So when we put our electrodes in and we record with electrodes along the tonotopic axis of the dorsal cochlear nucleus, and then we plot the spontaneous rate, this is what the spontaneous rate of a control group of animals looks like. So it's a normal spontaneous rate. But then what we do is we noise expose the animals, and this is showing the spectrum of the noise exposure, which is centered at 7 kilohertz, and it's a band pass filtered noise. We expose the animals to this sound very loud at about 100 decibels for 2 hours. And then we wait for the tinnitus to develop, and 8 weeks after 8 weeks post-exposure, we look and see what the spontaneous rates were. And in this case, we're looking at those animals that did not get tinnitus. So just as in people, noise exposure doesn't always produce tinnitus. It produces tinnitus in about half of animals. And so we have to look at what the animals' spontaneous rate that don't get tinnitus, because this is like a control for us. And so this is what the animals that do get tinnitus show. They show increased spontaneous firing rates above the noise exposure frequency. And this is one signature of tinnitus that is well accepted in the scientific community. So fusiform cells in animals with tinnitus show increased spontaneous firing rates. And this finding has been produced many times in our lab and also in other labs. Now, synchrony between neurons has been shown to be important for object perception. So you can think of tinnitus as like an object that you perceive. And so in order for us to perceive it, we not only need the increased spontaneous rate, but we need increased spontaneous activity between neurons. And so now we're going to show you what the cross-correlation coefficient between the neurons looks like, which is indicating the synchrony. And here we show the unexposed animals. This is the animals that don't get tinnitus. And this is the animals that do get tinnitus. And so we can say that fusiform cells in tinnitus animals show increased cross-unit synchrony as well as increased spontaneous activity. Now, I wanted to show you that when we look at the cochleas of these animals that have been noise exposed, and here what we're plotting is, this is ribbons per inner hair cell. So it's the number of neurons really that are affected in the cochlea after the noise exposure. And in this case, we exposed the left ears. And so we're looking at the right ears, which are in red, compared to the left ears that have been exposed. And you can see that these counts are slightly decreased in the animals that got noise exposure. Now, if we now plot the animals that got tinnitus and the animals that didn't get tinnitus, you'll see that there's absolutely no difference between them. And so because of that, we can say that tinnitus does not emerge in the cochlea. It's been well known for a long time that tinnitus emerges in the brain. So now we're back to our fusiform cell circuit. And I'm going to remind you again that the fusiform cells receive input on their apical dendrites at the top from the somatosensory system, and they receive auditory nerve input on their basal dendrites. Now, these synapses here between the somatosensory inputs and the fusiform cell apical dendrites are plastic. And I'm going to explain to you what plasticity is. But briefly, what it means is that if you stimulate these cells over and over again, you can change the excitability of this fusiform cell and its output to the central nervous system, to the further central nervous system. On the other hand, if we stimulate the auditory nerve fibers over and over again, they might show the excitation that I showed in the previous slide, but they'll come back to normal pretty quickly. And that means that these synapses are not plastic. So what I'm going to show you next is that activating these somatosensory inputs closely in time to the auditory inputs induces what we call timing-dependent plasticity. So don't worry if you don't get that right now, because I'm going to explain that very soon. But first, we'll go through what is long-term plasticity. And as I just said to you, repeated activation of a plastic synapse induces long-term increases in the excitability of the neuron. This process is called long-term potentiation. Now, we can also have repeated activation of a plastic synapse that can also depress excitability. And this process is called a long-term depression. So I'm going to use these terms long-term potentiation and long-term depression, or LTP and LTD, in the rest of the talk. In the fusiform cells, repeated activation of the somatosensory inputs and the auditory inputs in close timing can produce either LTP or LTD. And whether you're going to get LTP or LTD is going to depend on the relative timing and the order of the stimulation. So I'm going to show you now a little video of how this happens. So in this particular case, we want to induce LTP or LTD, and so we're going to stimulate the somatosensory inputs first, followed by the auditory inputs. And then I'm going to plot the spontaneous or sound-driven activity of these neurons when we did that. Now we're going to reverse this, so we're going to do auditory followed by somatosensory stimulation, and we're going to plot the spike rate for that. Now this defines a stimulus timing-dependent plasticity that we see in these fusiform cells, and it's because of the plasticity of the fusiform cell synapses with the somatosensory input, but combining the auditory and somatosensory input produces this timing-dependent plasticity. LTP and LTD. So here you can see that the neuron is more excitable, and here you can see that the neuron is less excitable. Does fusiform cell plasticity change in animals with tinnitus? Is our next question. And so to study this, we took a group of guinea pigs, and we first used a tinnitus test called the gap detection tinnitus test. We got a baseline. And then we divided these into an unexposed sham control, which didn't have any signs of tinnitus with our test, and a noise-exposed group, where we waited after 8 weeks, and then we tested them, and we showed that almost half of them, did not show tinnitus, and the other half showed tinnitus. So now we're going to do this experiment again in the animals with tinnitus. So in this case, we've got somatosensory before auditory input, and we're plotting the spontaneous or spike-driven rate. And then we're doing the opposite, auditory before somatosensory, and we're plotting that firing rate. And this is our normal control that you've already seen. But what happens with tinnitus? Well, you can see here that this curve is completely inverted. And what this means is that the circuit has changed. There's many reasons for why the circuit has changed. One of them being that there's an increase in somatosensory inputs, an increase in excitatory input in the animals with tinnitus. There's other changes like decreases in inhibition and changes in other neurotransmitters. But the end result is that this is an inverted curve, and what's more, there's much more LTP than there is LTD. And this is, again, another signature of tinnitus in the fusiform cells. So this increased excitability that we produced increases the output of the dorsal cochlear nucleus to the higher centers. So just to summarize here, we have this diagram of what I just showed you, and we can show here that what we're plotting here is the change in firing rate after we did the bimodal stimulation, and we changed the intervals between the auditory and somatosensory stimulation, as well as the order between the auditory and somatosensory stimulation. And for the normal animals, you see this white curve here. And then for the animals with tinnitus, you see that this curve is inverted and shifted up, producing more long-term potentiation and less long-term depression. In the animals that didn't get tinnitus, interestingly enough, there's very little long-term potentiation, and there's more long-term depression. So the circuit in the no tinnitus animals protected these animals from getting tinnitus. They didn't get LTP that made the neuron more excitable, but instead they had more LTD, similar to what we see in, not quite as much as the normal one, but more similar. So we can say that long-term plasticity can be induced in fusiform cells by combining sound with somatosensory stimulation. And we can say that fusiform cell plasticity is altered after noise exposure and further altered in tinnitus animals. Now, what I've shown you so far, we've been using deep brain stimulation to stimulate the somatosensory system. But if we want to use this for a treatment, we have to use a technique that's non-invasive. And so we tried to now do long-term depression induction using transdermal neck stimulation. And so our guinea pigs now, instead of deep brain stimulation, they got somatosensory stimulation on the neck. And that was combined with sounds in the same way that I've previously shown you. So here we can show again, for the normal animals, we got this kind of timing-dependent plasticity curve. For the animals with tinnitus, the curve was again inverted and shifted upwards, producing more LTP and less LTD. And in the no tinnitus animals, we see the same as what we saw with all of these curves as we saw with the deep brain stimulation. We see very little long-term potentiation, but we see long-term depression that is greater than in the tinnitus animals. And so we figured that if we want to reduce tinnitus, we have to induce long-term depression in the circuit, as seen here. And we have to do this over and over again and train the circuit to produce long-term depression and reduce the output of the fusiform cells to the rest of the nervous system. And so that is, in fact, what we did. We used these particular intervals and orders of auditory somatosensory stimulation to produce transdermally, and we stimulated a group of guinea pigs, and I'm going to show you that in the next slide. Again, here we took a group of unexposed controls. We tested their tinnitus first, and then we noise-exposed animals, and which produced half of them got tinnitus and half of them did not get tinnitus. And then what we did was we did bimodal treatment, the treatment that we showed to produce the most long-term depression. But we had several control groups. So we had somatosensory stimulation alone. And we know from our studies that somatosensory treatment can also produce long-term plasticity, but it mostly produces long-term potentiation. So we would not expect this to decrease our tinnitus. And then we did unimodal auditory stimulation, which I also told you that these synapses from the cochlea are not plastic. So we didn't expect this to have any effect at all. And what we did was we coupled this with an 8 kilohertz tone combined with transdermal electrical stimulation. The 8 kilohertz is important here because the 8 kilohertz matched the frequency of the tinnitus of most of the guinea pigs. And we wanted to see whether this frequency was important. Another reason that we picked the 8 kilohertz to be the same as the tinnitus was that this was going to target the pathological neurons. So the frequencies of the animal's tinnitus were going to activate those particular cells that were affected in tinnitus. We did this for 20 minutes a day for 25 days. So we can say from this that bimodal stimulation at specific auditory somatosensory intervals reduces tinnitus in guinea pigs. So this is showing you the conditions. We had the auditory alone, the somatosensory alone. Sorry, this was the sham. This was the auditory alone. And this was the bimodal treatment. And what did we see? So now we're plotting a tinnitus index, which is the amount of tinnitus that the animal had. And let's first look at where we saw no effect. So the shams that got nothing, just the same noise, the same environment, but they didn't get any stimulation. There was no effect on these. And the ones that got auditory alone are shown here in green, not too much effect over there as well. The ones that got somatosensory alone, you can see that a lot of them actually saw an increase in tinnitus, and some saw a decrease in tinnitus, but this was very inconsistent, so we wouldn't want to use this as a treatment. But now look at what happened to the animals that got the bimodal treatment. And this is important because when it was the same frequency as the animal's tinnitus, we saw a decrease in the animal's tinnitus. When it was a different frequency from the tinnitus, we did not see that. So it was important that the auditory stimulus was the same as the animal's tinnitus. So we can say, again, that bimodal stimulation at specific auditory somatosensory intervals reduces tinnitus in guinea pigs. Now, we also looked at the brains of these animals after we'd done these experiments. And so these are our conditions again. And this is what happened to the synchrony. And the same exact thing happened with spontaneous activity. And you can see that instead of having an increased spontaneous rate as these animals had when they had tinnitus, the animals that were treated successfully showed low spontaneous rates, whereas those that showed increases in the tinnitus index showed increased spontaneous rates and synchrony. And so the physiology and the behavior matched exactly. So now that we've shown that bimodal stimulation at specific auditory somatosensory intervals can reduce tinnitus and brain activity that reflects tinnitus, we ask, now what happens in humans? And so we did a first clinical trial, which was our pilot study. We recruited 20 adults with chronic tinnitus, and we chose for our first cohort of patients those that had somatosensory involvement in tinnitus. And that was those who could modulate their tinnitus with some sort of somatosensory movement that stimulated the somatosensory system. We chose normal to mild hearing loss for the pilot study, and we excluded patients who had retrocochlear pathology, for example, like a tumor on the eighth nerve, Lenire's disease, which is a cochlear disease, or drugs that affect hearing or the central nervous system, or tinnitus. And they couldn't have participated in a tinnitus study or treatment within 6 months of our study. We did exactly the same treatment that we did on the animals. So it was a bisensory auditory somatosensory treatment. Here's showing the electrodes placed on the neck of a subject. And the auditory stimulus was the same as the person's tinnitus, which, as I explained in the animals, is very important because we want to be activating those neurons that are pathological. So we're going to do transdermal electrical stimulation on either the neck, as shown here, or on the face. And this electrical stimulation was just above threshold, so the patients might feel it at the beginning, but then they couldn't feel it after that. The bisensory interval was derived from the preclinical studies. So this is the study design. There was first a baseline measurement of tinnitus, and we measured the tinnitus with an interactive program that the patients were put into a sound booth, and they had headphones on, and they were played lots of different sounds that they had to match their frequency and intensity of these sounds, as well as the type of tinnitus that it was. So it could have been tonal tinnitus, it could have been noisy tinnitus, it could have been chirping tinnitus. We consider all of these kinds of tinnitus. And so we treated the patients for 4 weeks, and we had two groups. So in sequence 1, the group got the active treatment, which was the bimodal treatment first. Then there was a washout period for 4 weeks. And then they switched over and got the sham, which was just auditory alone. Then the sequence 2, the groups first got the sham, and then they had a washout, and then they got the active treatment. And then after these, there was another washout, and then that was the end of the study. So what did we see? So what I'm plotting here at the top is a change in loudness, which we measured using the same iterative program that we had used to generate the person's tinnitus. And so what we're showing here in purple is the active treatment. This is the group 1. And you can see that there's a progressive decrease in the loudness of the tinnitus, which comes back after about a week to baseline. And then the second group is shown here, which shows exactly the same thing. It's a progressive decrease in the tinnitus and not quite a return back to normal. The shams, the ones who just got the auditory alone, showed very little decrease in their tinnitus. Now, we also looked at the change in tinnitus functional index, which is measuring the quality of life of the patient. And you can see here that the same trend follows that we saw with the loudness measurements. We see a decrease, progressive decrease in the TFI. And interestingly enough, this does not come back as quickly as the loudness change came back. But then we see the other group, we see the same decrease in tinnitus over time. And the auditory alone really also did very little, and in some cases showed an increase in the life impact of the tinnitus patients. So in the second clinical trial, given the success of the first trial, we recruited 99 adults with chronic tinnitus and somatosensory involvement, normal to moderate hearing loss, and the same exclusion criteria that we used in the first study. But now, the only difference really was that we used 6 weeks instead of 4 weeks in duration. And the treatment was exactly the same as the previous study. The auditory stimulus spectrum was the same as their tinnitus spectrum. And we used transdermal stimulation in the face or neck just above threshold. So what did we see here? This is what we got from the first trial. This is the study design. And this is the study design for the second trial, with the only difference being the 6 weeks duration. And also we did some measurements at the end of the trial up to 36 weeks. So what did we see? Well, I just want to go back to this slide here to show you that in our first trial, I showed you that after the treatment, the tinnitus came back to a baseline. Well, in the second trial, this actually didn't happen. And because we didn't get a return to baseline, we were only able to use the first half of the trial, because the second trial, which was a good result, was that there was such a long-lasting effect that we couldn't actually measure what happened because we still had an ongoing effect. So what happened here in this first phase of the study? You can see that as we plot the TFI score here. So you can see if you look at the purple, which is showing the treatment, you can see the progressive decrease that we saw in the first trial. But you can see that this is more dramatic than it was in the first trial. And we see this cumulative decrease in tinnitus. I'm looking at the TFI score now, the effect of tinnitus on life quality. So that at 6 weeks, we've got what's called a significant effect, because we get 13 points reduction in the TFI, which is considered the MCID for as treatment being successful. So you can see that at 6 weeks, we have this significant decrease in life quality. And when we look at 12 weeks even, we see that this is even more pronounced. And then when we measure at 28, 32, and 36 weeks, we can see that this effect is long-lasting, up to 36 weeks. So we can summarize this by saying that the TFI score at 6 weeks shows a 13-point drop, and that the cumulative treatment effect is shown, which demonstrates lasting durability of the effect. Now, we can also look at something that's called responder rate. And responder rate looks at the percentage of subjects achieving this 13-point TFI reduction. And so it's a 13-point improvement versus baseline in the active versus the sham group. And so what we're plotting here is the proportion of subjects that showed a 13-point decrease in their TFI. And you can see here, if looking up to 6 weeks, that this was at 65% in the patients that received the active treatment, but only at 25% of the sham achieved this 13-point reduction. So this is a pretty dramatic demonstration of the effectiveness of the treatment, which we were very happy to see. So now we also measured loudness, just as we did in the first study. And here we're looking at a loudness change measured in psychoacoustic, the psychoacoustic measure that we used with the interactive software. And for your elucidation, 6 dB change equals a 50% perceived volume change, and 12 dB equals a 75% change. So you can see here for the active group, shown in purple, that we got, at 6 weeks, we got 7 dB, so that's more than 50% of perceived volume change. And at 12 weeks, we got 11 dB, which is a 75% decrease in the loudness. So this is a cumulative treatment effect with lasting durability. So in summary, the clinical trial results showed that the second study replicated the pilot study, but with greatly improved longevity for loudness reduction and TFI scores. The treatment effect remained even after active treatment ended, suggesting long-term neuroplasticity changes of the type that I showed you in the preclinical studies. The treatment effect was so pronounced that subjects did not return to baseline during the washout periods. So what I have told you from basic science to clinical trial is that systematic studies of normal cochlear nucleus circuitry in animal models demonstrates long-term plasticity, long-lasting long-term plasticity in the circuit. And in our animal model that we had to develop to test the tinnitus, we were able to show behavioral and physiological evidence of increased spontaneous rates and synchrony. And in comparison with animals without tinnitus, this was a very large increase in spontaneous rate and synchrony. It's important to compare with animals that do not have tinnitus, because the animals without tinnitus can tell us something about resilience that is very important for future studies. I showed you that DCN circuit plasticity changes with noise exposure, and that DCN circuit plasticity further changes with tinnitus. This is important because if we hadn't divided the animals up into those that got tinnitus compared to those that don't get tinnitus, the whole effect could have washed out and that we wouldn't have seen anything. So that's a very important step to do, is to divide the animals into those that get tinnitus and those that don't get tinnitus in order to develop a treatment. And I showed you that auditory somatosensory stimulation to induce LTD reverses the tinnitus circuit in guinea pigs and in humans. None of this work could have been done with all of the tens of people that you see on the screen. The names in black are the people who participated in the 2 most recent studies, the preclinical trials with the animals and the human trials. But all of those orange or yellow letters showing all of the people over the many years have contributed to this work without which we could never have gotten to a treatment. So I thank you very much, and now I'll hand the microphone to Jon.
Thanks for that extremely informative presentation, Susan. Despite being your student now for 6 years, I always find when I listen to one of your talks that there are so many new things that I learn and insights that I gain. So I appreciate you being my patient teacher. Now that Susan has given a clinical and scientific overview of her therapy, I wanted to share part of our process in bringing her innovation from university research into the world of regulated medical devices. And as I talk about this process, my goal is to discuss our experience by being transparent, accurate, and hopeful. And I'll first touch on the opportunity that I saw when I met Susan in 2019, and then I will talk about the history and development of our device and our pathway to clearance in the U.S. market. Auricle is a venture-backed medical device company that has an exclusive focus, and that is to gain regulatory clearance for and then commercialize Susan's tinnitus therapy. The two founders, Susan and I, were introduced earlier, so I wanted to take this time to introduce the third founder, David Martell, Auricle Chief Innovation Officer. David is co-inventor of the therapy, and he worked with Susan at the Shore Lab during a 14-year period where he also completed his PhD in biomedical engineering. And at Auricle, David is one of the key resources that connects clinical insights into important device features. Back in 2019, when I first met Susan, I immediately discovered that this was an opportunity worth my pursuit, and there were three things that really stood out to me now as well as then. The first one is tinnitus impacts hundreds of millions of people globally, and sadly, there is no consistently effective therapy. Second, I was really impressed by the quality of Susan's research, and that convinced me that her therapy had a technical and clinical right to succeed. I believe that Susan has over 100 publications in total, with 33 of those focused just within tinnitus. And third, the $13 million in grant funding that Susan had applied for and received not only paid for the basic science and discovery and the early animal research leading to human trials, but it also paid for the development of 2 generations of prototype medical devices, as well as laying the foundation for 3 issued patents. So with all of this progress in place, it became very important for me to try to move her work forward with agility. Right from the beginning, I proposed a strategy to Susan and David that still applies today, and that is to maximize the utility of the work that had been completed and focus only on any essential gaps. And one of the ways that we have accomplished this is to engage the FDA via what's called the pre-submission process to really try and understand the most efficient pathway to clearance. So now I would like to talk about our medical device. On our left is our second generation clinical prototype device, and our handheld battery-operated system uses a single earbud for auditory stimulus and 2 electrodes that deliver mild nerve stimulation. And as we learned from Susan's presentation, the mechanism of action is to use these 2 modes to diminish tinnitus. And I like to think of her therapy as targeting and reprogramming the neural source of the tinnitus signal. After diagnostic evaluation in clinic, the device is taken home and used by the person with tinnitus for 30 minutes per day. And what you see here is clearly a prototype device, but I saw tremendous potential because we have a simple design, low cost of goods sold, and a clear user interface. But I realized very quickly that the totality of the system is not just what you see here. So, in order to understand that more, let's go back to 2019 and look at the complete device. This is the patient take-home treatment device that we just talked about on the prior slide, and it is connected to an audiologist interface, which is used to generate the person's tinnitus likeness spectrum. And this equipment is connected to a calibrated set of headphones, as well as a Windows PC that contains 2 proprietary software programs that are used to generate each person's customized treatment. And from a regulatory perspective, the entire system that you see here is considered our end-to-end medical device. It's not just the take-home treatment device that's shown on the left. So in order to understand the complexity of this system and the work that we needed to do, I thought it might be helpful to compare a university research prototype device with a submission compliant or ready device. So this chart compares a research prototype at the university level as well as a device that might be included in a submission on 6 different aspects. And if we look at the purpose of a research prototype, well, that is clearly to experiment or to test or to learn, whereas a submission-ready device is designed to convince regulators of a device's safety, reliability, and accuracy. From a user point of view, a research prototype is often just used by a few users and typically at a single site, whereas a submission-ready device is used by many different kinds of users at multiple sites and often in multiple countries. For hardware, research prototypes are often hand-built with many modifications and changes made along the way, but for a submission-ready device, the hardware must be manufacturing or production-ready and features a fixed design. Whenever a medical device contains software, research prototypes will usually use experimental code that may be buggy and require frequent updates, whereas a submission-ready device has a much higher standard. The software must go through a rigorous validation and verification process with versions that feature strict control. For research prototypes, documentation is often informal, written in things like lab notebooks. But for a submission-ready device, the record keeping is onerous and the document requirement is huge, and everything must be able to meet audit scrutiny. And finally, for regulatory compliance, research prototypes often feature little or no requirements because they are typically not intended for a major device submission. But for a submission-ready device, it must comply with all of the relevant international standards that apply. And just 3 examples of those would be ISO 14971 for risk analysis and assessment, IEC 60601 for safety and what's called essential device performance, as well as IEC 62304, which covers software development guidelines in medical devices. So as we understood the requirements for a submission-ready device better, that then led to our focus on creating a device that would be submission-ready or submission-compliant. So I'm very pleased to report that we have made excellent progress on exactly that kind of device, and I would like to preview that system for you today. Most importantly, our new system uses the exact same acoustic and electrical stimulation protocol that was used successfully in the pilot and feasibility studies. This means that we are delivering a customized acoustic and electrical stimulation protocol for every person with tinnitus. There is nothing that is pre-programmed in our device. Also, we now have a single box solution which combines the patient take-home treatment device with the audiologist interface in a single, elegant solution. So this means that in clinic, the system is connected to a computer and is used to generate the treatment protocol for that patient. And then the patient takes the device home and uses it for home treatments. We are also rewriting the software and coding it for real-world clinicians and patients. And of course, all of this device development and design work is being done according to the relevant and applicable medical device standards. So, how will we bring this device to market? I would next like to talk about the regulatory pathway. Despite the work that we have completed on the system that I just showed you, we need about 16 more months of device development time. And that covers bench testing, verification, validation, and all of the electrical and safety testing that needs to be performed. When that work is complete, we will have our first opportunity to determine if we will make an FDA 510(k) submission. And this would represent our fastest pathway to a clearance. But on the other hand, if we determine that additional work is required, and that could be in the area of, for example, clinical data collection or other performance testing on the device, then that could certainly add months or quarters to the process. We are not able to predict what that might look like because we are not at that point yet, but I can absolutely assure you that during this entire process, we will be making decisions working closely with our regulatory, clinical, and device development experts, and we will always be balancing speed, risk, and cost. And I certainly understand and know that these device development timelines can be disappointing to many, but to speak truthfully and candidly, we are trying to balance the desire to bring a therapy to patients as rapidly as possible, but we are also constrained by regulatory requirements as well as capital requirements. So in summary, even though we still have a lot of work to do ahead of us, we see the finish line in sight. And I thought that it'd be nice to close on some very positive points. First, let's remember that we have 2 completed human trials that have shown compelling safety and efficacy. Second, we have made tremendous progress on the design and development of a new system that uses everything that has worked, but at the same time improves reliability, usability, and meeting regulatory requirements. And third, we have engaged the FDA as a partner, not an adversary, and we have keen and detailed understanding of the regulatory pathway as well as the criteria for success. So thank you very much for your interest and time today. We are excited to respond to the many questions that I am certain will come in, but we're especially interested in hearing from those of you that are living with tinnitus. Your perspective matters deeply to us, and the work that we are doing is dedicated to bringing a solution to you.
All right. Thank you so much, Jon and Susan, for your presentations. We'll now go into the Q&A. Again, we are live together from California. Thank you also to everyone in the audience who's joining us live today. I see we're having a very active chat discussion, which is great. I saw a few comments about the lack of closed captions. We will be adding those in the final edit, but many of you should also have the option to create live captions on your device if you wish to do that. All right, we will go into the questions and answers. But I want to ask first a question directly to you, Jon, because I suspect this one will be top of mind for at least those people who have been following your and Susan's work for the past few years. And that's regarding the timeline, because many might have expected or hoped that the timeline would be shorter. So I just wanted to give you the opportunity, and I know you did address it already briefly in your talk, But I just wanted to give you the opportunity to expand on that a bit and maybe tell us a bit more about how the timeline evolved and some of the decisions that you had to make along the way.
Sure, I appreciate that question, Hazel. Thank you very much. I think I'll state that, first of all, we too share the frustration that medical device timelines are long and complex. And I think a lot of this is driven by the fact that it's a challenge whenever you're taking a university developed novel innovation and bringing it into the highly regulated world of medical devices. That's just kind of a reality that we have had to contend with. I think that we can speed up the process by the application of resources to the program. That's one of the things that is, of course, a requirement, is to be able to have effective capital resources to move a program forward. So in any opportunity that we're able to do that, we certainly will. So we're trying to do the best that we can and move as quickly as possible, but we are a venture-backed device company. And so that's sort of, where we are today with it.
All right. I'm sure some of the questions will also be touching on this, so let's not dwell on it now. Let's start actually pulling up some questions from the audience. And the first set of questions we'll be addressing have been pre-submitted, or rather synthesized from hundreds of pre-submitted questions. And the first one is, when will the device be released in which countries? So US, Europe, etc., different countries. And what's the current status of FDA approval or other regulatory processes? You did already touch on it, but can you explicitly answer that question?
Yeah, I think my timeline that I presented in my talk did speak to the U.S. market, so I probably don't need to cover that further. In truth, we are focused exclusively on the U.S. market right now, and that really just comes down to resources and personnel and what we can do. We certainly do recognize that 97% of the population lives outside of the United States, and certainly the need and interest and requirement for a tinnitus therapy is huge around the world. We are designing the device so that it will comply with international requirements, so that when we have the resources to address markets like the European Union, Korea, Japan, other places, I think we'll have a device that will at least be ready for that. And I think that if we have strategic interest and involvement in the company at some point, that would definitely solve a lot of the resource application challenge. So at this time, we are just focused on the U.S. market. But again, that is really personnel and resource driven.
All right. Let's move on to the next question. And guys, you can keep submitting questions in the meantime in the Q&A box, but maybe first check if someone else has already asked your question and then upvote that one. Okay, this one is very direct. Why is it taking so long? Will you keep us informed about the current status more regularly in future, rather than keeping everything private and going radio silent?
Yeah, I think the fact that it's taking so long, the simple answer is really, when you are taking a novel university-developed innovation and you are trying to bring that into the regulated world of medical devices, that process is extremely long and cumbersome and challenging. And I think that I've been at this for just 6 years. Susan has been working on this for over 20 years. So she is probably the most impatient and the most interested in having her therapy come to market. But I think it really does come down to all of the work that is involved in the development of a medical device. It's interesting because I think that a lot of people believe that we have already answered the most difficult questions. Is the device safe? Is it effective? And that has been demonstrated in the human trials that we have done, which is great news, but it really does take a lot of time to satisfy the regulatory requirements that are behind medical devices. And I would argue that a lot of that is a good thing. We want our regulators to be approving devices that have demonstrated safety and efficacy, because that gives then patients confidence that those systems are cleared for use.
Could you address maybe also the communications aspect of the question? Can people expect more regular updates going forward?
Yeah, in truth and honesty, I think it's very challenging for a medical device or a drug company to provide regular updates with what they are doing. Part of that is based on the fact that you don't want any communications to be seen as sort of pre-marketing of your device. But another part of it is just the uncertainty in the regulatory timelines involved. The regulatory consultant that we work with, she has over 40 years of experience in devices. But I think even for her, it's extremely difficult to try to forecast and determine how an agency will respond to a different submission. I think that in my experience in working with the FDA, it's not a digital process where questions are either yes or no. A lot of the communication with the Food and Drug Administration comes down to your ability to construct cogent arguments based on data. And oftentimes you are acting as an advocate. So you are representing positions, you're trying to explain things to regulators. So all of that makes the process a little bit challenging to forecast. And so I think that if we attempt to provide updates to the marketplace, we absolutely want to avoid disappointment that can cause. So I think that we will have some degree of updates that we will provide when we have relevant, important, actionable news. So that's sort of my feeling about that.
Okay. So if I'm summarizing this, you will communicate about milestones that you have achieved, but you don't want to set the wrong expectations by making, predictions about things that are uncertain.
Right.
Okay.
I think that's
Just paraphrasing you there. All right. Okay. Yeah. Let's go on to the next question, and it is: will the device only be available, in audiologist offices, or can patients take it home after initial fitting? Will frequency testing also be possible at home? Where are the electrodes placed? Okay, it's actually a series of questions, but mostly it's related to, can it be used at home? I guess it's more of a Susan question. Is that right?
We could probably share this.
Yeah.
Yes, at first the device will only be available going through an audiologist. And the reason for this is because, as I explained in my talk, the auditory part of the stimulus, it's that the person's actual tinnitus. And so that has to be measured. And as Jon showed yesterday in his talk, the eventual device will have the machinery to do all of the audiologist testing so that the tinnitus spectrum can then be placed on the device to be combined with the electrical stimulation. And then the next part of the question is, yes, the patients will be able to take the device home after the initial fitting, and they will be able to have the device that's programmed according to what the audiologist has found with the initial interview.
Anything to add, Jon?
Yeah, I think that's a great answer, Susan. So thank you for that. I would offer an example, and I think that the example of hearing aids is perhaps one of the best ones, because when hearing aids were first developed, those were only available through typically an audiologist who would do the fitment and the sale of the device to the person needing it. But then in 2016, Congress passed what is called the OTC, or the Over-the-Counter Hearing Aid Act. And then that allowed hearing aids to be sold not just through an audiologist or a professional, but also directly to a consumer through other channels. And I think that for our device as well, we envision perhaps a similar pathway. As Susan explained in the beginning, the device will have an FDA clearance as a physician-prescribed home use device. So that means a healthcare practitioner, in our case, typically an audiologist, would be the person that would be doing the diagnosis and the fitment of the device, selling it to the patient to be used at home. But our product roadmap does envision in the future a version of the device where fitment could take place in the home environment, and then a person with tinnitus would be able to purchase the product directly. But in order to do that, we have both a regulatory aspect. So, for example, we would need to be able to go from a physician-prescribed home use device clearance and add what is called an over-the-counter clearance to our set of clearances. And then also there are some technology challenges in terms of being able to ensure that fitment for a device could take place in a home environment that is safe and accurate and effective.
Right. So it could be a possibility at some point in the future to make it fully or almost fully at home, but it won't be the case from the beginning. It's fiat. All right. Okay, let's move on to the next pre-submitted question. And in the meantime, feel free to keep submitting more questions. Okay. This is probably more for Susan. So do SSRIs, those are certain class of antidepressants, I believe, or other psychiatric medications reduce the efficacy of the device? And why were patients on SSRIs excluded from the trials? And can people taking these medications still use the device?
Okay, certainly. So I'll start with the last part of the question, and that these medications were excluded from the trials because they themselves can produce some changes in tinnitus, and usually by making tinnitus louder or even inducing tinnitus. And so if we had included people in the trials that were using these medications, then we wouldn't really be able to assess whether changes in their tinnitus were due to our treatment or whether they were due to the medications. And so that's why that was an exclusion criterion. The question about, whether the medications would interfere with the use of the device, I don't think that would be the case, because we would expect the same kind of effects on the person taking the medication circuitry as with other people. So it really was an exclusion just to make sure that the trial was done in a very rigorous manner.
Yeah, that sounds encouraging. Did you want to add anything, Jon?
No, I think
Okay.
that was
That was
great.
covered. Okay, then we will move on to the next question. Can the device also help with related conditions such as hyperacusis, dysacusis, tonic tensor tympani syndrome, or visual snow syndrome? And is it possible to use the device with these conditions? I'm just finishing the question there.
Okay.
Yeah.
Well, the treatment that we've developed was based on an altered tinnitus circuit. And it wasn't based on testing these other conditions, for hyperacusis. So I would say no, that it probably wouldn't help these other conditions because it wasn't designed specifically on the circuit that the different circuit might be involved in these other conditions. That said, there is some commentary in the literature that hyperacusis and tinnitus share some similarities in that they often co-occur with each other.
Yeah.
But in our work in the lab, we have some findings that suggest that hyperacusis is really governed by a different circuit, and that's initiated in the ventral cochlear nucleus and not in the dorsal cochlear nucleus. So just thinking about the two different disorders, tinnitus is a disorder of spontaneous activity. It's a disorder in the absence of stimulation, whereas hyperacusis is a disorder at the opposite end of the auditory stimulus, in that it's a disorder of high intensities, and it's a sound-driven disorder. So while there might be some overlap in the occurrence of these two disorders, at the moment, I don't think that their mechanisms overlap sufficiently that our device would also help with hyperacusis.
Okay, makes sense. But so what if a hyperacusis patient who also has tinnitus wants to use the device for their tinnitus, but they're worried about, putting the sound directly in their ears? Is there an alternative for that? Can they listen to it through a speaker, for instance?
I think that the answer to that really is going to depend on the intensity of the stimulus. So people who do have hyperacusis are afraid of loud sounds. And the intensity that we present the stimulus is really governed by the patient. So if they find it to be uncomfortably loud, then we can turn it down. So I think it would be okay for people with hyperacusis to use the device to treat their tinnitus.
Great, wonderful. All right, let's move on to the next question. Okay, how effective is the device overall? I think that was more or less addressed in your presentation, but maybe a quick recap. And then who benefits the most? And are the results permanent or temporary? And why do some patients not respond? Again, it's a number of different questions grouped together, but basically about efficacy.
Well, we did show in several ways that I presented in my talk that the tinnitus effect on life and as well as the loudness decreased significantly over the 6 weeks of the treatment. And then we looked at how much of a decrement in the tinnitus did a certain percentage of the population exhibit. And we showed that we took a criterion that was 13 points in decrease in the TFI, which is a clinically significant decrease in the TFI. And we showed that for the active treatment, 65% of patients showed that clinically significant decrease, whereas only 25% of people using the sham showed that decrease. So that is a massive difference, and I think that really speaks to the efficacy of the treatment, as well as showing that when we looked at the loudness, that after 6 weeks, patients saw more than a 50% decrease in their tinnitus loudness. And at 12 weeks, they saw up to a 75% decrease in their loudness. So I think if I had really bad tinnitus, I'd be pretty happy with that.
Is there anything you can say about the responders versus non-responders, and are there any distinguishing characteristics that you were able to observe?
Well, yes, when you talk about responders, there's another question there is like, what do you mean by responding? And I think many studies, when they talk about responding, they mean, did this person show any decrement at all? Whereas we're using a very strict criterion here of 13 points reduction. And so we can say that we have a very significant, statistically significant and clinically significant reduction. But we could also look at which patients showed less decrement than 13 points. And when we reduce the criteria to say, 1 or 2% decrease, there were only 3 patients in the whole study that showed no improvement at all. And so I think that's a great result, because those patients who didn't show a clinically significant decrease of 13 points might have shown that if we had continued the treatment for even longer. So this is something that we need to test in the future.
All right. So you might, if you're able to do further studies, would you be interested to look into why some people respond better than others?
Well, I think that, yes, could be looked into real data. And with bigger populations, you might be able to see other factors that this kind of patient might benefit more than the other kind of patient. But so far in our population, we haven't identified that length of tinnitus or loudness of tinnitus or frequency of tinnitus, or any of those are causing differential results.
Okay, good to know. All right, well, just checking if you wanted to add anything to the past question or two.
Yeah, I think, Susan, that was a fantastic answer. What I would maybe add to that is I am often asked a question, especially by investors, what is the ultimate course of treatment? And the honest answer is we don't know what the ultimate course of treatment is. Our 2018 study featured 4 weeks of daily 30-minute treatment. The 2023 study increased that period by 50% to 6 weeks of 30-minute daily treatments. And what we saw from those 2 studies was a much improved durability of treatment effect from that longer treatment time. So that does seem to indicate that a longer treatment period, or at least an intensive treatment period, does create more of an effect, not only in terms of the effect of the treatment, but also the duration. But I think in the real world, the real question is, what will happen when the device is cleared? How will people use it? And a lot of that we just don't know today. So I think that has implications for the early commercialization part of the device and market, where we can try to understand those questions better.
Great. So I see the next question has already popped up. Can it also help with non-somatic tinnitus, low frequency hum, or broad hissing sounds? I think the non-somatic is, of course, relevant because your model of tinnitus hinges on the somatosensory input. And I think were there any exclusion criteria actually that people who don't have a somatic tinnitus couldn't take part? I'm trying to remember.
Yes,
Yeah.
as that was actually our first criteria.
Yeah.
We felt that at the outset, at least in our first clinical trials, we should choose people who had known somatosensory involvement in their tinnitus. But having said that, the animal studies that we've performed, for example, if we deafen an animal, we show that there's somatosensory infiltration of the cochlear nucleus, and this happens in 100% of the animals. And so that makes me optimistic that even though somebody might not be able to modulate their tinnitus, there still is somatosensory involvement in their tinnitus, and they would benefit from the device too. But we haven't tested it, and so we don't know.
Okay, but that's potentially encouraging. And the other part of the question was about is hissing and low frequency sounds. Is there anything you can say about that?
Yes. So when we test for the person's tinnitus, we provide them with a variety of kinds of tinnitus sounds. So that would include hissing, it would include chirping and something like cicadas, and it would include more tonal kind of tinnitus too. So those would automatically be included in this assessment.
Great. All right. Let's move on. I think we still have a couple of the pre-submitted questions to tackle. How is stimulation calibrated and tinnitus frequency determined, and how precise must the match be? And can the device handle multiple or changing tones? I think this is again for Susan mostly.
Yeah. Okay. So as I alluded to in my talk, the actual auditory part of the signal is the person's tinnitus. And the way we assess that is with an interactive computer program where the person puts some headphones on and they're played sounds, a whole bunch of different kinds of tinnitus-like sounds. And they first pick what kind of tinnitus they have, is it hissing or tonal. And then they're played a number of different sounds of different frequencies and different intensities. And they keep on matching this until we have a good match to their actual tinnitus. And then that tinnitus is then transposed to be the auditory part of the stimulation. So what was the next part of the question?
I was the hissing and low frequency sounds, so I think you've covered it.
Right.
Yeah.
Oh, and then the calibration.
The calibration. Yeah.
Yeah. So that's sort of how the calibration of the auditory stimulus takes place. And then the device itself is limited in terms of the volume output that it can be. And the sound part of the stimulus is calibrated according to the person's comfort. So it has to be within a certain intensity range, but it also has to be comfortable for the patient.
Yeah.
The somatosensory stimulus is calibrated as in the electrical stimulation on the skin. And it's calibrated so that the person has to detect the electrical stimulation. And then what we do is we set that electrical stimulus level to just below the threshold. So they first feel it, and then we set it to just below the threshold. So they're not actually feeling it. And the reason for it being sub-threshold is not only for blinding, but it's to adhere to similarity with previous studies that were shown in vitro recordings that showed that the phenomenon of spike timing dependent plasticity was really most effective when the somatosensory stimulus was sub-threshold.
Oh, interesting. Yeah, I think there's another question still coming up that also touches on this, but let's just see what the next question is. How does Auricle differ from Lenire or other bimodal stimulation devices? I know you guys don't want to talk about specific companies or people, so you can answer the question in a general sense. And the second part of the question is, why was Auricle chosen over larger medical manufacturers as the development partner? I think I'll first go to you, Jon, for this one.
Okay. Yeah, I would say that there are certainly a plethora of neurostimulation, neuromodulation, bioelectronic devices that are available globally to treat the symptoms of tinnitus. I think the thing that really separates our therapy from those of others is the scientific basis. And Susan certainly spoke to this in her presentation. I touched on it as well. But if you examine her 33 peer-reviewed publications just within tinnitus, you can really sort of see the arc of her research, because she started not with the question of, how do I treat tinnitus? But she really started with, let me understand multisensory integration. What is the source of the tinnitus signal in the auditory pathway? Let me test that in a validated animal model, and then I'll focus on how to modulate that signal. And all of that information is publicly available, so it is certainly available for critique and review. And I do think that that is a huge differentiator for our therapy, because there is a strong basis for it. I think another component that is equally important is I've learned that tinnitus is a highly subjective condition where the placebo response is significant. And this is very similar to pain or dizziness or anxiety. And so when you are dealing with a chronic subjective condition like that, study design becomes absolutely essential. So you need studies that have an active treatment group as well as a control, and they have to be double-blind, randomized controlled trials, because you really need to understand what is a placebo response versus a treatment effect. So I think that for a person with tinnitus who is looking for some sort of a therapy, those are probably some important questions to ask and examine and look at. So I think that it's certainly not enough to just claim that you have a biostimulation device. What's really important is the science behind it, the protocol that is used for the treatment. Why are you using an auditory stimulus? Why are you using an electronic stimulus? What is the order, the duration, the interval of that? And so I think those are the things that really kind of separate, I believe, the therapy that Susan has developed.
Do you want to touch on the second part of the question, which is actually a whole separate question, actually?
Yeah, I forgot to cover that one as well. I get asked this question quite a bit too, and typically people will ask, why is it that this effort is being supported by a venture-backed company like Auricle? And I think that the main answer to that is, at least in the context of the United States market, if you are looking at university innovation, the typical pathway for that to go from university to commercialization is through solo entrepreneur-led efforts. And often the larger universities will have offices of technology transfer, or in the case of Michigan, they call it the Office of Innovation Partnership. And that office's prerogative is to identify individuals that they will often hire into positions called a mentor in residence or an entrepreneur in residence. And those people often have entrepreneurial experience, and they work with researchers like Susan to try to guide opportunities along. And eventually, if they find an opportunity that they become interested in, they'll often take it out of the university into a startup. So that is really a typical model. The way I approached it was I was not an entrepreneur in residence or a mentor in residence at Michigan. I just discovered the opportunity of the university IP model. And I made an appointment with the director of the Office of Tech Transfer, talked about my background and interest in home use consumer devices. And then that's when he made the introduction to Susan. So I think that is more the typical way that innovation leaves a university and goes into markets. In terms of larger companies, why hasn't a larger company approached this and put their resources behind it? And again, this may be more typical of the United States, but larger medical device companies will typically get into these sort of opportunities at a much later stage. So rather than at the stage of early research or early IP development, animal research. That's typically not the stage that the larger companies will come into. They prefer to have products that are much closer to commercialization. And that simply means that those products will then be immediately revenue accretive to the company. Having said that, I do think that strategic partnerships are valid and very important. And I've actually presented to several of the global hearing healthcare companies. They are all not only aware of Susan's research, but keenly interested in what we're doing. But my sense is that they would prefer to become involved once we have a regulatory clearance in a major market and are close to selling. So I hope that answers the question.
Yeah, that makes sense. So it's just not like the big pharma companies were jumping at the opportunity, unfortunately, to take you guys to market. So you had to find your own way there.
It's true, but I do think there's a lot of advantages to this model because we are able to be agile. We can remain true to the scientific and translational vision. And I think that it prepares us well in the future for partnerships that might come up.
Yeah. Susan, do you want to add anything to this?
No, that
Okay.
was perfect, Jon.
Sounds good. Then we'll move on to the next question. All right. Yes, of course, we get an AI question, as you must these days. With the rise of artificial intelligence, could an AI be integrated to make it a more user-friendly device? And I would add to that question, maybe, is there any other potential application of AI that you might see with this device? And I'll start with you, Jon.
Yeah. I will provide a very general answer first, but I think we all know of, the incredible interest in artificial intelligence in so many different areas. I read a statistic the other day that said that in the first half of 2024, I think venture capital investment in AI represented about 20% of total investment dollars. This year, if you look at the same period, I think that has jumped all the way to 70%. So I think it's an indication of how important and interesting AI is for a lot of different areas. I wish that we were able to say that we were AI driven or AI centric, but we're not. But I do think that AI may have a place in our device in a future iteration. I think that specifically for the patient fitment process of the device, there are several ideas that we have that could make that faster through the implementation of large data systems.
Sounds good. Susan, anything? No? All right. Then we will move to the next. I think it might be the final one of the pre-submitted questions. And also, I believe this question is, oh no, it's a different one than I thought. Fine. I thought we were going to do the Dan Polly one. All right. Which tinnitus types or causes doesn't the device work for, and who shouldn't use it? Oh, we kind of touched on it already, but Susan, is there anything else you can add? Because I feel like we covered it more or less, but
We did, yeah.
all right. Yeah, I think this was covered in an earlier question. So we will go to the next one. Yes, so this was the one I was expecting, and this is a pre-submitted question, but unlike the other ones, which were kind of like an aggregation of dozens of questions, this one is from a specific person, and that person is Dan Polly, a professor at Harvard, who's also part of Tinnitus Quest. And I will read it from my phone because on the screen the question kind of gets cut off, and I want to make sure I read it properly. So Dan's question is, how was the electrocutaneous stimulation intensity determined? Would stronger stimulation improve therapeutic effects? And for blinding, could both groups receive this stronger stimulation with the placebo group using a timing interval that shouldn't induce spike timing-dependent plasticity?
Mm-hmm. Hello, Dan. Good question. I think I have answered this partially in a previous question. So I've mentioned that the cutaneous stimulation intensity was set to the threshold, iteratively determined with the patient, and then it was set to just below the threshold. And the reason for that was for blinding, of course, and also because the previous basic science studies in vitro that originally demonstrated spike timing dependent plasticity used subthreshold somatosensory stimulation or parallel fiber stimulation. But your question is a good one in terms of, so I don't think, just to follow that up, I don't think increased intensity would make this more effective if we're looking at the rules governing spike timing dependent plasticity. Your suggestion of using different timings is a good one. And this is something that we actually wanted to do, but in our interactions with FDA, they wanted us specifically to compare bimodal stimulation to auditory alone stimulation. So in the end, that's why we chose that. But I think for future clinical trials for blinding, I think your suggestion is a good one. And one would have to work to figure out the exact timing intervals that we would be sure wouldn't be causing any plasticity.
Yeah, I think that's really useful additional information about like the FDA requirements kind of restricting you a bit there, that this could still be an interesting test setup for the future. I think we've covered all the pre-submitted questions, so the question now is what questions were submitted live, and my colleague Marco has been moderating them, and we're going to cover the ones that are really new or different compared to what we've already covered. And the first one is, what might explain why participants who received audio-only treatment first had no volume reduction of tinnitus when they later received the active bimodal treatment after the washout? All right, so this is to do with the sequence of either first receiving the sham or first receiving the active treatment, if I understand correctly.
Okay, yeah, it's a little difficult to understand the question, but what I think this person is asking is, what's the difference between whether you receive the active first or second? And for the 6 weeks treatment, I can't really answer that because, as I explained in my talk, the 6 weeks treatment was so effective we really couldn't do the second. We couldn't analyze the second half because there was still ongoing processing during the time that we would have been presenting the second sequence. But in the first study, where we did 4 weeks, we didn't really see any difference between the first sequence and the second sequence, and it didn't really matter whether you got the active treatment first or the sham treatment first. We got the same results.
Okay, yeah. And then thank you, by the way, Harley, for that very good question. We will pull up the next one, unless you wanted to add something, Jon.
No. I on it.
All right, then we will move on. And I really don't know how many we're still going to get at this point, because it depends how many questions you guys submitted that were actually new compared to the pre-submitted ones. Is the treatment ineffective if you have tonal tinnitus at 11 kilohertz or 13 plus kilohertz, since Tin-Tester doesn't generate tones with those frequencies? And maybe you can also explain what bit about the Tin-Tester and what you use that for.
Okay, so the Tin-Tester is the iterative computer program that I've mentioned several times already. And I don't know where the person got that information for, is that it doesn't generate 11 kilohertz or 13 kilohertz. It definitely does.
Okay.
And it's multiple tones of many different frequencies are presented, because almost no person's tinnitus is absolutely pure tone. It's I've never heard of tinnitus that's absolutely just one tone, which would make it a pure tone tinnitus. It can be tonal, so it can have the quality more of a tone than as hissing, for example, which is sort of more noisy. But in both cases, so let's say you had tonal tinnitus, then the first phase of this iterative program would be to choose whether you have tonal tinnitus. Does your tinnitus sound more like these tones, not tone, but tones? Or does it sound more like hissing, or does it sound more like chirping? So you first pick that. And then once you've picked that category, it presents multiple different tones combined with each other, different spectra of tones, until you find the match to your tinnitus. And then it gets closer and closer until it produces tinnitus.
Is there any types of tinnitus that are extremely hard to match, or do you always somehow get a match?
I think we've had good success in matching people's tinnitus.
Great. All right, then we will move on. And that question was also from Harley Ryder. So thank you again, Harley. Okay. In the second study, the audio-only group did not experience benefit after switching to the active bisensory group. Why? This sounds like maybe the same question as before, but I don't know what your interpretation is, Susan.
It is, but I'd actually have to sort of look at the graph in front of me to try to understand the specificity of the question.
Yeah.
But we didn't expect to see an improvement with audio only. And the slight improvement that we saw in both studies, we attribute to a placebo effect.
Okay, makes sense. All right, let's move on. Okay, is there an upper limit to the frequency of tinnitus tone that is treatable? 8 kilohertz, 16 kilohertz, 20 kilohertz? I think from what you just said, there's no limit?
No.
Okay. I think the short answer is always good. No, the answer is no. Okay, there we go. Did any patients see their tinnitus worsen permanently after using the device? Yeah, this is obviously also always something that people do worry about with any kind of treatment.
Yes, of course. So we didn't see any patients that used the active treatment for the active treatment that showed any worsening of their tinnitus that was related to the device. We did get a couple of people whose tinnitus worsened, but they were using the sham device and not the active device. And then we had a couple of people, 2 or 3 people whose tinnitus got worse after they did something like go to a rock concert, and they reported that to us, and or they had to take ibuprofen because they had some illness. So
right. Okay. So you could basically rule out the possibility that it was in any way caused by the treatment itself.
Right.
That is reassuring. And that question was from Vic. Thank you very much, Vic. And let's see what else we got. From JP, can you go over some of the main protocols for daily life alterations while going through the treatment? Were loud sounds to be avoided, stimulants like caffeine or ADHD medication, the rest cuts off, but I think you get the idea. Yeah, what would you say to that?
Well, in the trial, our subjects were instructed to dedicate this half hour to their tinnitus treatment and go and sit in a quiet room, which was important because we don't want other sounds interfering, and basically just sit and do the treatment for 30 minutes without really doing anything else. We didn't say, you can't do this or you can't do that, but when patients reported back to us, that's what they did. They sat quietly in a room and they did this for 30 minutes. So I think if you're reading something, maybe it's okay, but I would say it would be best to sit quietly and just do this treatment for 30 minutes.
I do want to ask Jon if you have anything to add, because it's also a bit of a commercialization question in a way, because any treatment that you bring to market, it has to be like feasible for the patient to do that and incorporate it in their daily life. How do you look at that?
Yeah, I think it's an excellent question, and it really does illustrate the difference between an extremely controlled environment, which a clinical study typically is, versus compliance and how people with tinnitus, once they have the device in the marketplace, will actually use it. Clearly there will be differences. So I think that commercialization implication there is when the device has clearance, I don't think it's a great idea to just rush into the market and try to sell as many as possible. But I think that a phased approach to the market is appropriate so that we can understand a lot of the real world scenarios and questions about how a device that has been successful in a clinical study is actually used in the real world, because there will clearly be differences.
And I expect you intend to sort of follow that real world implementation and experience to learn from it.
Absolutely. I think that, we have a lot of questions that we touched on earlier about the ultimate course of treatment. And I think our suspicion is that there will probably be a period of intensive device use until a person with tinnitus achieves a satisfactory reduction in loudness and quality of life improvement, and then perhaps some sort of a titration beyond that point. But again, this is just speculation. This is something that we need to understand. And so I think that work is actually very important.
Absolutely, yeah. And thank you, I think it was JP, for that question. And we're going to see if we have any more. Yes, we do. If lowering the volume of the audio stimulus due to hyperacusis, does this reduce efficacy of the treatment as long as you can still hear the treatment audio at low volume? Again, from Harley. And I do think it was covered, Susan, but do you have anything to add still?
I don't think it was.
Okay,
Yeah,
okay.
that's a good question.
Okay.
Because, is the intensity of the stimulus important for the
Right.
treatment? And within a range, the intensity is important. The most important part of it is that the person hears it. And so if the person with hyperacusis lowers it to a point where they can't hear it, well, then it's not going to work. But if they lower it to just a level that's comfortable, then it probably would be fine.
Okay, that's reassuring. And thank you, Harley, for yet another very good question. Keep them coming if you want. I don't see any other. Oh, there we go. Will the device work on tinnitus that is only modulated by very specific movements, such as yawning, but otherwise does not seem somatic?
Yes, well, we defined any movement that produce a change in the tinnitus as being somatic. So
Yeah. So it's a matter of
have-
definition, because people have different definitions for somatic tinnitus.
right, it didn't. We did prefer to have people who we could measure it very easily. But if somebody shows that they, yawning produces a big change in their tinnitus, well, by our definition, they have somatic tinnitus.
They have somatic tinnitus, right? And from my understanding, that is actually the majority of the tinnitus population that can modulate their tinnitus in some way. I
Yes,
think
absolutely.
60-70%.
Yeah, 2/3 of it.
Yep.
Well, yeah.
Yeah, exactly. Yeah.
I mean, you have to show them how to do it. So there have been some studies that have gone out into people, asked people in the street, can you modulate your tinnitus? And they said that
What do you mean? Yeah.
I think you have to explain it and show them how to do it, and then you'll find the true percentage.
Absolutely. The next question is from Melissa. Do you believe that this could be equally as effective for patients who have suffered from tinnitus for a long period of time versus patients who have been recently diagnosed? And I believe you said you saw no difference there, but can you confirm that, Susan?
Yes, we didn't see any difference between people who had it for. We just did specify more than 6 months.
Oh, yeah.
We didn't test people who had tinnitus for less than 6 months because we wanted to make sure it was chronic tinnitus. But given that, if the person got it this year or they got it 20 years ago, we didn't see any difference.
Yeah, that's good to know. And thank you, Melissa, for that question, which comes up anytime we talk about tinnitus treatments. The question regarding multiple frequencies at once hasn't been answered in that previous response. If someone had multiple frequencies at once, would the device not work? From Paul.
I think we answered that and said that nobody has frequencies that are not multiple. So everybody has a spectrum, and nobody has just a pure tone.
Yeah. But in essence, the device works regardless of whether you have a narrow band of frequency or a wide band or whatever it is. Yeah.
Right, as long as we've matched the person's tinnitus, because the reason for using the person's tinnitus as the auditory stimulus, as I showed in the animal studies, is that this is targeting those neurons that are producing the tinnitus. And that's why we want the sound to be like the tinnitus, because that's the pathological region.
Yeah, makes sense. And thank you to Paul for that question. We will move on to the next one. Oh, that's very brief one, but I'm sure one that's on many people's mind. How much will the treatment cost? And of course, I'll look first to Jon.
Yeah, sure. Happy to answer that. The short answer is we have not made any determination about what the cost of therapy will be. And that's because we're not in market yet. But what I will say is that, as I mentioned in my presentation, the device itself is extremely simple, not only in design, but in parts. There's an enclosure, there's 2 printed circuit boards. We just have standard components on it. So if you look at all the traditional elements that go into a device's cost of goods sold, we have a lot to work with here. And my personal preference, and I believe that Susan and David both feel this way as well, is we want to make the therapy available to as many people with tinnitus that would like it. So that is our position on at least making the device available at a cost that is reasonable. I think that another part of the pricing question, it really comes down to the channel of distribution that you select. So in a very simple example, if a company has a product that they are selling directly to consumers, they usually have tremendous flexibility in being able to make pricing decisions. But if you have a product or device that you are selling through a distribution channel that involves a healthcare professional, well, then obviously you have to take into consideration the dynamics of that channel. And so, for example, audiologists will have certain expectations about how they would prefer to price a device to a person with tinnitus. And a lot of that really does depend upon how much time they're having to spend with those people. And so, for example, if an audiologist is finding that they're spending a lot of time with a person with tinnitus, not only at the fitting, but post-sale, then those are things that tend to drive up the cost of therapy. So I think that, those are parts of the pricing equation that we certainly have a big influence over, but not complete control over. But we do intend to make the therapy available to as many people as possible at a reasonable price.
And this also comes back to partly to that question about at-home use versus going through the audiologist's office. And if you can find a way to make it more self-serve, let's say, it would by definition be cheaper.
I think that's right. I think that, again, the example of how hearing aids are distributed and sold in the United States is a great example. For people that feel more comfortable going to a professional, an audiologist, to have a fitment, that works very well, and they will tend to pay a price that is higher for that service. But for people who are able to do an assessment online, they have a higher priority perhaps for a lower price point, then a direct-to-consumer hearing aid might be appropriate for them. So I don't think that even in our product roadmap, when we have a device that would be available for patients to fit and purchase directly at home, that doesn't necessarily eliminate the professional channel. I think that, we would definitely have both of those channels because people with tinnitus will have different needs and different preferences.
Great. Let's move on to the next question. And again, I don't know how many we still have coming. The timeline in the presentation was not clear. Okay. What part of 2026 is the most optimistic submission to the FDA?
I tried to not put exact dates into the presentation. I did have years and quarters or months just specified to sort of give a general indication of what that timeline could look like, because trying to talk about a specific year, a specific quarter, a specific month, I think it provides a false sense of precision in terms of what we truly know today. So I tried to outline 2 different possibilities, and the fastest possibility would be to put together a 510(k) submission as soon as our device development work is complete. As I mentioned, that is about a 16-month process. So if we determine to file an FDA 510(k) submission at the end of the device development, then we could have a clearance in hand much sooner. But on the other hand, if there is additional requirements, and this is really going to be determined through close interaction with the Food and Drug Administration in terms of our device, the arguments that we put forth, then that could add significant time to the timeline. And again, I think that, there is a very understandable desire to try to pin down an exact year, quarter, month, but it's just not something that not only me, but none of our expert consultants are able to do that. We're just working as fast as we can.
Yeah, all right. Thank you for that elaboration, Jon. And thank you also for the question. I forgot who submitted it, but thank you. And we will go to the next question. Does the average loudness change in trial 2, plotted in purple in the slide Jon's loudness change, include or exclude the loudness changes for the people who dropped out of the study, I imagine? Wow, I have trouble bringing to mind which slide exactly this was. Susan, can you answer that?
I do, but honestly, I don't remember which one I showed. So
Okay.
I would have to go back and look at that.
We will skip this one.
Yeah, I can answer a little bit on that. Whenever you are looking at a clinical study, and especially a pivotal study for FDA submission, you're always looking at your enrolled patients split between what are called the intent to treat group, and that is the total number of enrollees in a study. And so in the Jones study, that was 99 subjects. And then you look at also the group of patients that were able to complete all phases of the study, and that's the per protocol group. So we had 99 in the ITT group, 56 in the per protocol group, and we did show loudness reduction data for both of those groups in the study.
Mhm.
Yeah, right.
As to whether that specific slide was one or the other, I would have to go back and look.
Mhm.
Okay. Well, thank you, Eric, for that question. The next one is from Luca. Can sessions be longer than 30 minutes or treatment be longer than 6 weeks? Are there any limits? Susan.
Yes, no, they can't be longer than 30 minutes at the moment, because we've determined that to be a good duration based on the efficacy of the treatment, as well as factors like how long can a person sit still. And so these durations weren't just thrown out there. They were considered through discussions with audiologists and physicians and tinnitus patients to determine what the best compromise would be. Now, in terms of efficacy, if we did it for 3 hours, I think maybe it would be better, but we don't actually know that. But in terms of what's realistic, that wouldn't work, because nobody's going to sit there quietly for 3 hours. So we came up with 30 minutes as the optimal amount of time for this treatment. Now, in terms of longer than 6 weeks, well, we showed that you got a much better result with 6 weeks than with 4 weeks. And so it's very likely that if we went to 8 weeks or 10 weeks, we'd get an even better result. And this makes sense in terms of the underlying mechanisms of the treatment, in that it's long-term plasticity. And when we're retraining the circuit, if you do it for longer, you get better results. So in the future, there wouldn't be a limit to how long the person could use that, or this would be determined in the future in real-world data.
Makes sense. Jon, do you want to add anything? Because I imagine there's also potentially regulatory aspects to here, or other things where, you have to stick to the protocol as it's prescribed.
Yeah, that's exactly right. I think the thing I would add is that Susan's explanation was perfect. The device also has a built-in limitation so that it can only be used for 30 minutes in a 24-hour period. And so that is the way that the device is designed. And in terms of, longer than 6 weeks, I think this does get back to a discussion we had earlier in this conversation, which is, what is the ultimate course of treatment in the real world? And I think, as Susan alluded to, at least in the differences that we saw between the Marks and the Jones study, that additional 2 weeks of treatment really did seem to have tangible durability and treatment efficacy impact. So it seems reasonable that a longer period of intensive use might also extend that effect. And perhaps for people that were not responding at the 13-point reduction in TFI, a longer treatment period might bring them down to that. But again, I think, these are questions that we will be very interested in investigating post-clearance.
Yeah, with real world data, he said.
Yeah.
Exactly. Okay, yeah, thank you, Luca, for that question. The next one is from Grogu. Can the bimodal interval, the interval between electrical stimulation and sound stimulation, be customized in some way to each patient in order to achieve more effective LTD?
Well, it's a very interesting question. I think that would be very difficult to do. Because how would you even do that, since the effect is taking, the treatment is taking a while to work? I think that would be very difficult to do. And we did it the other way around, whereas we took the data from the preclinical data from the experiments that we did on animals, where we could extensively vary the interval and the order, and we could look at the actual neural responses to those different intervals and orders. And so, what I showed you in the summary slide was that this 5 milliseconds, it was a summary slide, and I didn't show you the underlying data, which showed that most of the neurons that we looked at individually showed that 5 millisecond interval to be the most effective in producing long-term depression. So I think that is a more accurate way to determine that interval than to try to do it on an individual patient without really having a plan of how you would actually measure that.
It seems we're receiving more sciencey questions than you had maybe expected, but good questions here from the audience. And thank you, Grogu, for that question. Here's another one from Harley again. Is it possible that efficacy could be improved by using audio and electrical stimulation on both sides at the same time for each treatment session?
Yeah, that's an excellent question. Mm-hmm. And it's one that would need to be determined in the future, because in our preclinical studies, we used only one ear. And in some of our extended preclinical studies where we tested individuals, individuals reported that even though they have an earphone just in one side, it affected their tinnitus on both sides. And that is, of course, because tinnitus is generated in the brain, but it can be lateralized to one side or the other. So it is possible that that the treatment effect would be different if we used bilateral stimulation. But I think, again, this is something for future studies to determine. Good question.
Yes, thank you. Thank you, Harley. I heard we still have a couple of questions coming up before we start wrapping up, and we've been going for about 2 hours now. Thank you also for your endurance, by the way. Could this be beneficial for reactive tinnitus, and what is currently understood about the underlying mechanisms of reactive tinnitus? Yeah, that is an interesting question, Susan, because I think there's no consensus really on if reactive tinnitus is a separate thing, or maybe it's hyperacusis, or I don't know if you have an opinion on that.
Well, I would ask this question to define reactive tinnitus before I could answer the question. So maybe he'll send or she will send in another question.
Yeah, I assume what the person means is that your tinnitus gets aggravated by certain environmental sounds. So some people do report that. Yeah, that's typically what we hear from quite a few people. And as far as I know, in academia, there's no clear consensus on that phenomenon and whether it is a distinct form of tinnitus or maybe it's actually hyperacusis.
Right.
Else. Yeah.
So I'll have to say that I don't know the answer to that question.
Okay. That's also always a very good answer when you don't know. Are there concerns that other promising treatments, such as electrical stimulation of the cochlea, could crowd out investment in the Auricle device? From Michael Alford. Wow, Jon, what do you say?
Yeah, that's a great question. I don't believe so. I think that there are so few tinnitus therapies that are actually really promising and effective today. And we're certainly hopeful that there may be more, perhaps on the drug development side as well. But I think that in terms of the work that we're doing, we're pretty far ahead of, I think, a lot of competitors, just simply because of the amount of time that Susan has spent on her research. And I think that that demonstrates that if you really are trying to develop a therapy that has a sound scientific basis, that you have gone through animal studies in a validated animal model and then gone on to human trials, that work takes a tremendous amount of time. So I'm certainly hopeful that there are other promising therapies that receive funding, but I don't think that they or us would see one another as competitors for dollars.
Yeah, I mean, I would also say the more the better. Certainly, since we pretty much know for sure that no treatment will work for everyone. I mean, that's one reason, and you want to certainly hedge your bets. And the more investment in tinnitus, the better, I would think. And that's why we
Sure.
also created Tinnitus Quest in an effort to support the emergence of new ideas on how tinnitus could be silenced. And I think the more ideas and the more treatment development, the better. Yeah. Susan, have anything to add?
No, absolutely. I think the more the merrier of good, sound treatments. That's better for tinnitus sufferers.
Absolutely. Yeah. So thank you very much, Michael, for that great question. From William, you indicated that bringing this to market is subject to capital requirements. How much is Oracle looking to raise, and how can we help? Jon.
Fantastic question. Yes, we definitely require capital to move forward. We actually have an investment round that we just started now. So that is a $3 million raise that will fund the activities that I had outlined in my presentation. And in fact, we have a very short video that we will be showing as part of this interview that will provide some of those details. So thank you for the question.
Yeah, we will indeed come back to this topic after we've covered the final questions from the audience. I think we have maybe a couple more coming up. No, actually we don't, because it says, thank you, everyone. The Q&A session has now concluded. So that means there were no new questions, or only questions that had already been covered. Yeah, and that was my cue to go to the funding topic, Jon. So maybe you can say a little bit more about like what strategy you're employing to raise new capital, and just how you're going about it, before we then play the video that you guys created to help raise the capital.
Yeah, sure. Thank you for that opportunity. As I just mentioned to the person who asked the question, we do have a funding round. This is actually our first equity round. So that means we are selling preferred shares. The raise is $3 million, which is very much in keeping with the way I have been building this business. We have a very capital efficient approach to how we are raising and deploying funds. And I believe that the video will be instructive in providing information, because it will show a URL link landing page that will answer a lot of the basic questions about the investment. The investment is only open to what are called accredited investors. That is also defined on the landing page when you see that. And we do have an investment minimum, but I'm hopeful that people might find that of interest. Because one of the things that was very interesting to me about our company is all of our individual investors that are accredited are people with tinnitus. So they very much believe in Susan's work and the work that we are trying to do to bring her wonderful therapy to the market. So it does seem that there is big interest from people that, suffer from this terribly debilitating condition.
That is good to hear. And yeah, we will in fact now play your video before we wrap up this event.
Music is your heartbeat. Sound is your craft.
Are you ready to make some noise?
But what happens when sound turns against you? As artists, we live by rhythm, resonance, and voice. But years of loud performances can leave a lasting mark. A relentless ringing, buzzing, or roaring that never stops. This is tinnitus. When your brain hears a sound that isn't really there. It affects over 740 million people worldwide. For 120 million, it's life-disrupting. DJs, singers, actors, veterans, factory workers, anyone exposed to loud environments is at risk. Tinnitus can lead to anxiety, isolation, and in severe cases, despair. And when tinnitus gets loud, it doesn't just hurt. It isolates, depresses, and in some cases, devastates. Current solutions only mask symptoms. Nothing treats the root cause. Until now. Meet Auricle. We're developing the first home-use medical device that treats tinnitus at its source. Invented by Dr. Susan Shore at the University of Michigan. Her breakthrough is backed by 20 years of science, spanning animal research to human trials. It retrains the brain with targeted sound and gentle electrical pulses, fit by an audiologist, then used daily for 30 minutes at home. In trials, 65% of subjects saw meaningful improvement, compared to just 25% using a sham device. Loudness dropped by 50% in 6 weeks, and up to 75% by 12 weeks. Safe, simple, and scientifically backed. To bring this innovation to the world, we're raising money to complete device development, plan clinical trials, and secure new patents. All of our current individual investors suffer from tinnitus. They know the need firsthand. Join a mission with massive global impact and real potential returns. Auricle is building a future where tinnitus doesn't control your life, because we believe relief should be real, lasting, and accessible. Let's stop masking the noise and start silencing it at the source. Visit auricleinc.com/join our mission to learn more and invest today.
All right, everyone. Well, that about concludes this event for today. I want to thank Jon and Susan so very much for being here with me today in person and taking the trouble to travel here. And I do think it made this so much more engaging and interesting, certainly from my perspective, even though challenging to organize. And before we wrap up, I just want to ask each of you if you have any concluding remarks to share with our audience. And Susan.
Well, Hazel, I just want to thank you again for organizing this event. And I think it was very gratifying for both of us to be part of this. And, I hope that it gives some clarity to the many tinnitus sufferers who have been constantly contacting us. So I hope there's some comfort there, at least in the clarification and the hopefulness that I feel and I hope is transmitted to them as well.
Thank you, Susan. And Jon?
Yeah, I echo Susan's comments, and I think most importantly, I wanted to thank Tinnitus Quest for putting this on, and especially you, Hazel, for all of the work that you and Marku have done to organize this. There's actually a crew of people here that I'd like to thank as well, because I know that the setup for these kinds of events can be extremely challenging. I, too, am hopeful that the content was useful and informative to the many people living with this condition that are very interested in Susan's research and have been following her for years. And I hope that we achieve the right balance of transparency, clarity, as well as having hope. We are extremely excited about this innovation and really bringing it to the world. We certainly wish that it were a faster process, but I think we have extremely good understanding of what it takes to bring it to market. And so we're excited to be able to provide updates in the future as well.
Thank you for your kind words, Jon. And finally, I would like to thank our audience for tuning in today, for asking so many great questions. I do hope you all got something useful out of it or interesting. Tinnitus Quest will be organizing more live Q&As in future, and some of them might even be in person like today. So we very much hope to see you there. We'll round it off with a final video, but thank you again so much for being here today, and we hope to see you at our next live Q&A event. I never knew how important silence was until it was taken from me.
Now I hear noise in my head 24/7.
My doctor said that there's nothing to be done. People tell me just to get on with it.
But not all of us can.
Tinnitus Quest aims to revolutionize tinnitus research and speed up the quest for a cure.
Through quick, agile proof-of-concept studies.
And using trial and error to iterate on and improve potential solutions.
We know that more can be done to help tinnitus sufferers more quickly.
And cost efficiently.
We already have commitments from several big donors, and we have some of the best tinnitus researchers on board.
If you care about silencing tinnitus, donate to Tinnitus Quest.
We promise to communicate with you frequently on our progress, both the ups and the downs.
Because the road may be winding,
but we will get there in the end.
Shh.